My job is to make college easier because students have a lot of fun.
Hi students, I hope you are all good and doing well. Hi students, I hope you are
It's Professor Dave. Let's talk.
Okay, before I will proceed to your this exercise, let me first introduce to you about what is spectrophotometer. Okay, so I will stop share muna and share another window. Okay, spectrophotometer is principle and the very slow. Okay, so in order for us to easily understand that, so that we can have an overview about how can we use the UV spectrophotometer and also how is it important to the determination of concentration of uh, certain substances, not just alcohol, but also many compounds also can really detect absorbency.
Spectrophotometry. He knows a lot about the science stuff. Professor Dave explains. As we have learned in a
Can you, hear the sound?
Yes, ma'am.
Okay.
Yes, ma'am.
tutorial,
I will.
Kinet. It's Professor Dave. Let's talk spectrophotometry. He knows a lot about the science stuff. Professor Dave explains. As we have learned in a previous tutorial, kinetics is the study of reaction rates. Sometimes we will want to know how quickly or slowly a chemical reaction will happen. Therefore, any kinetic study will require monitoring the concentrations of a particular reactant or product over time to see how quickly they are being used up or formed. Of course, we can't see molecules and count them. They are just too small and too many. So to do this, we have to be clever. We can measure the mass of a precipitate as it is forming, or we can measure the volume or even the pressure of a gas that is produced over time. But what if a reaction does not produce a product in a different phase than the solution? Luckily, one of the most reliable methods involves using spectroscopy, which is the study of how light interacts with matter. So let's learn about this technique now. All molecules absorb and emit light differently based on what kinds of covalent bonds are in the molecule. If the products and reactants interact with light differently enough that there is a color change as the reaction occurs, we can monitor this process with an instrument called a spectrophotometer in order to measure changing concentrations. This will give us data in the form of an absorption spectrum, which displays how much light through a band of frequencies is able to pass through a sample to reach the detector. Where there are peaks, it means that not as much of the light of that wavelength is getting to the detector, which means that it is being absorbed by the sample. If we collect data like this continuously over the duration of a chemical reaction, we can gather information about how rapidly the absorbance changes, and thus how the concentration of a particular substance is changing over time. In order to do this, we will use an equation that relates the amount of material in a sample to the absorption of light passing through it, and this is called Beer's law. Here, A is for absorbance. This symbol represents something called the molar absorptivity, a measure of how well a chemical species absorbs a given wavelength of light. B represents the path length, or the distance light has to travel through the sample, which is relevant because the more of a substance the light travels through, the more of it that will be absorbed. And C is the concentration of the sample, which again is relevant because the more concentrated, the more absorbance that will occur. Absorbance is measured by comparing the intensity of the beam of light as it enters the sample and the intensity as it leaves the sample and strikes the detector, which are I0 and I respectively. And A will be equal to the log of I0 over I, or equivalently, the negative log of I over I0. If we plug the latter version into the original equation, substituting for A, and then solve for I, we can get this version of the log, which displays the exponential relationship between variables that are easily measurable. The primary advantage of this technique is that we can just let a reaction occur normally and measure the rate indirectly by analyzing the light that interacts with the components in the solution, rather than stopping the reaction and measuring concentrations directly.
Excuse me, ma'am.
Try
Yes.
one calculation to put this
Oh,
in context.
kuan mang nagbarina, ma'am ba?
Kung ano, medyo koan, disturbing.
Di mi ka kuan sa video, ma'am.
Ha?
Di mi ka bati sa video, ma'am ba?
Padumog.
Say that a substance has a molar absorptivity.
Kana,
Usapaw siya.
musapo siya. Okay.
Pero okay ra, ma'am, ang kuan, ma'am. Okay ra ang video, ma'am.
Oo. Sige. Namugod ng kuan eh. Sige lang.
Of 0.35 inverse centimeters inverse molarity. Light passes through a 2 centimeter sample and hits the detector with an intensity of 0.12 compared with the incident beam. What is the concentration of the sample? If we want to use the more basic version of Beer's law, then first we need to calculate the absorption. We can use this simple equation from before, plugging in 1 for the initial intensity and 0.12 for the other intensity. That gives us 0.9208 for A. Then we will use Beer's law. We can rearrange to solve for C, our unknown, plug in the absorption we calculated, plus the two given values, and we should get around 1.3 molar for the concentration of the analyte. Gathering this kind of information regarding concentration and plotting it in real time using computer software will allow us to easily infer all kinds of things about the rate law and hence the reaction mechanism for the reaction that is happening in solution, which is incredibly useful data. To get just a bit more practice, let's check comprehension.
Okay, let's go
As the
back.
reaction is count them.
As you have class that the absorbance, so this is the use for determining concentration of a given solution. Okay, when the product itself is still it's homogeneous also to the liquid, so it's not a form of gas or a form of solids, but then it's just the same in the solution. So with
They are
this
just
kind of instrument class, so it uses light absorbance. Okay, so it is basically it's how much light that's able to pass through the solution. Okay, meaning absorb ang iyang light. So i-absorb na siya. So just like ethanol is a liquid form. So and then also, diba, when we want to drink something alcoholic drink, so there are certain percentage of alcohol is present, diba? 4% alcohol, 5% when we flavored beer, ganun. So that is why using this spectrophotometer, you can really determine its concentration using this absorbance. Okay, and then also, so that's the principle of the Beer's law that the light source is zero, and then also how much light is absorbed. So sa passing through the solution. So meaning there are other instances class that that your solution is with color. Okay, you can also use this spectrophotometer in different chemical reactions with color, mga colorimetric, because it's really effective and also accurate in measuring concentrations with different colors, because different absorbance will really be given by the instrument when there is different different uh, granulation of colors. So you can really determine its concentration, how much is absorbed to this, ganyan, ganyan. So that is why here in the ethanol determination. So since ethanol class is colorless, no, so that is why there are certain wavelength that is set in the spectrophotometer in order for you to determine the ethanol. So the highest peak. So there are certain wavelength. Okay. So I will now share to you about how to measure concentration, or how to use the spectrophotometer. Okay. Mabati ra class.
Yes, ma'am.
The spectrophotometer is an instrument used to measure the effect of a sample on a beam of light. We can learn a lot about a substance by studying the way it interacts with light. Here's how a spectrophotometer works. A lamp provides the source of light. The beam of light strikes the diffraction grating, which works like a prism and separates the light into its component wavelengths. The grating is rotated so that only a specific wavelength of light reaches the exit slit. Then the light interacts with the sample. From this point, the detector measures the transmittance and absorbance of the sample. Transmittance refers to the amount of light that passes completely through the sample and strikes the detector. Absorbance is a measurement of light that is absorbed by the sample. The detector senses the light being transmitted through the sample and converts this information into a digital display. Let's practice by performing a simple experiment. In this exercise, we'll determine the absorbance spectrum of red food coloring. To begin, set the wavelength to 380 nanometers. With the sample chamber empty and the lid closed, set the display to 0% transmittance by using the zero control knob.
Okay. So based from my experience class is that dili na siya knob ang itutulog nimo. It's just meron na kang meron ka lang pipindutin na is equals to zero, which is very convenient na lang. So with the latest model of spectrophotometer, and also if you there are different brands to that, and there are also different cuvettes, different multi container of cuvettes na pwede kang magdagay ng sample na marami na. So, yun ang, but here is a classic equipment. So, the basic equipment for the spectrophotometer. So,
There are two cuvettes. One contains only milli-Q water. This is our blank.
so blank class is that it contains the distilled water. You can use distilled water, but there are other laboratories. It's what I have experienced from the UPLB. We have used the ionized or triple distilled, in which there are no ions into it na talaga that can interfere with the absorption of light. It can give absorbance because there are compounds that really can absorb the light. So it will really give absorbance. It will have a reading of absorbance. So it is really preferable that you have to use mga triple distilled. There are no traces of metals or ions that are present in the um, blank sample. Okay, so that's your reference point.
Wipe the blank cuvette with a Kim wipe to remove any fingerprints, and place the cuvette into the sample chamber by aligning the guide mark.
So this is the sample chamber class. So here in this equipment, so that's show man yung cuvette, but the cuvette, there are many kinds of cuvette. Then gagmay lang yun siya kaayo class na mamaya I will show you later. So gagmay lang yun siya na apoy plastic, na apoy glass. Okay? So there are two sides of the cuvette in which the opaque side, in which you can just hold that the opaque side of the cuvette, while on the other naman kay very transparent and very clear because it should be parallel to the when you place the cuvette into the sample chamber, you have to place the clear glass parallel to the source of light, to the light source, okay? So that this will be passed through. This will be used as the light will really pass through the sample. So not the opaque side because really it will not detect absorbance, no, because that is so that's the use of the opaque and also the clear glass. The opaque is in order for you to hold your sample. You should not touch the clear glass because it may also interfere for the reading of the absorbance. Okay, and also you use Kim wipes, it is a specialized tissue because it will not give you or it will not leave traces unlike other ordinary tissue that ang uban kay muligog ka ng mga debris of tissues, but for the Kim wipes, it's really specialized only for the this spectrophotometry, in which it is also expensive. So, kaya yun, mahal talaga ang Kim wipes. Ito lang yung na-encounter ko na tissue that is really expensive. So, should not be used in your face because, yeah.
Marks.
Okay, so let's continue.
Adjust the display to 100% transmittance. Remove the cuvette with the blank solution. Now set the display mode to absorbance by pressing the mode control key. The second cuvette contains Millicue water and red food coloring. This is our sample. The absorbance of the sample is 0.845. Press the mode control key to select transmittance. The transmittance for the sample is 14.2%. Increase the wavelength by 25 nanometers to 405. Wipe the blank cuvette again and place it in the sample chamber. Adjust the transmittance control knob until it displays 100%. Now remove the blank. Wipe the sample cuvette and place it in the sample chamber. The transmittance for the sample at 405 nanometers is 7.4%. The absorbance for the sample is 1.130. Record this in your lab notebook. Increase the wavelength by 25 nanometers to 430. Repeat these steps with the blank and sample writing each value in your lab notebook. Increase the wavelength by another 25 nanometers to 455 and test the sample again. By repeating these steps up to 680 nanometers, we can plot the absorbance spectrum for red food coloring. Now that we've finished our first practice exercise using the spectrophotometer, you're ready to enter the lab and try it for yourself.
Okay, so that's uh, the demo and the practice experiment. So as you can see that as you have watched from the demo experiment is that increasing ang iyang wavelength because I think there are many cases that you have to identify what if you are not sure of the wavelength of a particular compound that you wanted to detect its concentration, you have to play around the different wavelengths in order for you to get the uh, correct and accurate concentration. But if you have already a reference, because hindi naman siya ibig sabihin na that particular wavelength that's really um, the compound, the specific compound is really absorbed. So, hindi siya masyadong efficient unlike many other techniques or purification techniques that have really a sharp and really very high resolution of detecting specific compounds like the HPLC, the UPLC. So, ano ito? So, it has drawback pa din. So, it is not really specify just particular wavelength, pero ano din talaga siya, there are many disadvantages about it. So for the demo experiment, so he wanted to know on what wavelength does the food coloring has the highest peak. Okay? So, and then if he located that, di ba, na i-plot dito on what particular wavelength within the absorbance. So, that will be the wavelength that he will use for the different concentration of food coloring found in the in your containing in your solution. So that's it. Okay, so I will now. Is there any question, class? Is it clear? Class, are you still there?
Yes, ma'am.
Is there no question?
None so far.
I will now share again the laboratory exercise in which I will post this. So, hindi ako naka-PowerPoint ngayon. So, I will just give it to you to this word. Okay, so the title for your experiment or exercise, virtual exercise is spectrometric determination of ethanol. So this is a modification method by Sir Andresito Acabal, who was a professor at the Department of Pure and Applied Chemistry here in Visayas State University. Um, I think we have baka na abutan niyo siya sa DOPAC. So he just recently retired. So ayan. So the objective of this experiment or exercise is to determine the levels. Wait lang, class. Is to determine the levels of ethanol present in alcoholic drinks and ethanol produced from immobilized yeast fermentation setup. So, using the spectrophotometer, you have to determine its concentration through absorbance. So, meron din siyang uh, calculation of this, no? So as you plot your absorbance over your concentration, you have to determine, you have a standard curve to analyze also. Okay, so here in this procedure class, so prepare 50 ml of 2, 4, 6, 8, 10, and 12% volume, percentage by volume from absolute ethanol. So this is your standard concentration. So 2%, 4%, 6%, 8%, 10%, 12%. In which ang total volume sa imong concentration, each of your concentration is 15 L. Okay? I assume that you know how to do it already, no? So wait, volume by volume. So when we say if you prepare 100 ml, then you wanted to have a concentration of ethanol is 2%. So pila man ang imong volume sa imong ethanol nga isagol sa alcohol. Kung 2% ang imong i-prepare, 100 ml. Class. Pila man ang imong kuhaon nga volume sa ethanol, nga sa absolute ethanol. If you want to prepare 2%, then ang total volume is 100 ml. Pila man, class? So you have 2 ml of absolute ethanol and then ang imong tubig is 98 ml. So kung 50 ml man ni siya, ang inyong i-prepare is 1 ml ang inyong absolute ethanol, then 49 ml ang imong tubig. So that equivalent to 2% of 2% ethanol. Okay, so the same calculation that we will have the 4%, 6%, 8%, 10%, 12%. So kung 4%, 50 ml, no, you have 2 ml of your absolute ethanol, then 48 ml of your water. So you have to reflect this in your calculation in your lab report, how to prepare these solutions. So ako na magihatagan og sample ani. So yeah, you have to prepare this standard concentration. So you have to analyze and calculate this because in the face-to-face kamo dyan mag-calculate ani class. And then also prepare different dilutions, which is 2x, 5x, 10x. What do you mean by this? Okay, when you say alcoholic drinks, Like for example, San Miguel flavored beer. So you have to, the San Miguel flavored beer 10 ml, but you have 10 ml of San Miguel flavored beer, which is usually 4% medyo na ang kanang concentration sa ethanol. And they have 3%, 4%, ganon. So, when we say 2x ang i-dilution class, imo nang dublihon ang total volume sa imong sample. So, 10 ml, so meaning 20 ml na siya. So, imo dungagan o another 10 ml ang imong solution ka ng para mahihang 2x dilution sa imong San Miguel flavored beer sample. So, 2 times diluted na siya. So, because 10 ml of, you have also 10 ml of your water. So, it's dilution. If you say 5 times, so, ka-5 mo kapiloon ang 10 ml. Okay? So, na siya ang 5x and 10x of alcoholic drinks. The sample must pass through activated charcoal before dilution. So this is a way of purifying your sample so that there will be no interference during your spectrophotometer reading. Yeah, and then also you have to transfer 10 ml in 25 ml volumetric flask containing 12.5 ml potassium dichromate. And you have this calculation now that you have to weigh 3.768 grams of potassium dichromate and dissolved in 100 ml of distilled water with 325 ml of concentrated sulfuric acid and dilute to 1 liter. Then you have to place the flask in water bath at 62 degrees Celsius for 20 minutes. Okay, so cool the room temperature and dilute to 25 ml. So after that, after the water bath, you have to allow to cool it and then dilute to 25 ml. After that, use sample concentration and also your standard concentration. Pipette 5 ml into test tubes containing 5 ml distilled water and then shake in order for have even distribution. And then after that, after dilution, okay, you have to read the absorbance at 578 nanometers in the UV visible spectrophotometer. Okay, it depends upon where I will show you what the cuvette is look like. Yeah.
My name is Ariel, and I just started my first job as a project manager. When I was a student,
First, let's go over the parts of the spectrophotometer. First, you have the sample compartment. The lid opens and closes to put your sample.
Okay, class, I will show you again another video so that you will know the parts of the spectrophotometer.
Sample inside. First, let's go over the parts of the spectrophotometer. First, you have the sample compartment. The lid opens and closes to put your sample inside. You have the wavelength control knob to change the wavelength. This is called the zero control on the left-hand side. And this is the transmittance absorbance control on the right-hand side. Right on top is the mode select button. And when you push the mode select button, you'll notice that the LED will change positions. And so will the readout. To begin using the spectrophotometer, you have to have your samples ready. So first, you should prepare a blank solution. And the blank solution has everything in it except for the molecule or compound you wish to measure. Then you should also have a tube which contains a sample which you want to measure. When taking readings, it's best to go ahead and use the transmittance as the way to set up the machine and also to take your readings. The transmittance scale is linear, so it's easy to set the 0 and the 100%. So when you're ready to calibrate the machine, be sure to push the mode button several times until you have the transmittance value highlighted before you begin. In order to start taking a reading, you have to first set the machine up. So to do so, first thing you must do is use the wavelength control knob and adjust the wavelength until the wavelength that you want to measure at is reached. And let's go ahead and try taking a measurement at 420 nanometers. Next thing you need to do is, using the 0 control, turn the 0 control until you get 0.0 showing up on the display. Next thing you should do is now open up the sample compartment and put the blank solution in. You should always wipe off the bottom of your cuvettes before you insert them in the machine to make sure that nothing interferes with the reading. Also notice that there's a little line shown in the front of the cuvette. Line that up with the dash on the front of the sample compartment, and then insert it all the way inside, and then close the sample compartment. This time you're going to use the absorbance transmittance control and turn it appropriately one way or the other until you get 100 on the dial. Once you've set the machine at 100%, then you can go ahead and take your cuvette out and then put your sample in. Again, making sure that you line up the mark on the cuvette with the mark on the sample compartment. Here you can see that the transmittance value is 93.0%. You can also take an absorbance reading by simply pushing the mode button to go down to where it says absorbance. So when the absorbance LED is lit, you can see that at the same wavelength, the absorbance is 0.032. You want to ignore that. The machine has to be set up specifically in order to use that option, and this one is not. So do not write this concentration down, and you need to know the correct factor in order to set the concentration. So ignore those. Just use the transmittance and absorbance on the machine. When you remove your sample, the machine should return back to 0 transmittance. You can go ahead and continue to put new samples into the sample compartment and continue to take readings at the same wavelength that you just set the machine up for. So if you have 10 test tubes, you can now put all 10 of them in one at a time and note the readings. But once you decide that you want to record a wavelength at a different wavelength, let's say we want to switch from 420 to 480 nanometers, now we have to go through the entire calibration method one more time. So then we'll have to begin by first changing the wavelength to the desired 480. We'll have to 0 the machine using the 0 control knob on the left. Then we'll have to put the blank cuvette back in. And then we'll have to use the knob on the right, the absorbance transmittance control, in order to set the machine back at 100% transmittance. And then after that's complete, then you can go ahead and put your sample in to read the absorbance or transmittance at the new wavelength.
Whoa! Adapted Mind Math! Come explore the site with me. I'm in first.
So, cuvette, no. Okay, class, this is a, a photo of the cuvette. Okay, so sa kanang inyo ang na-watch ka ganina, ang cuvette ato is more test tubes na dagko. But then here, kadi siya, more on siya nga gamay ng mga 1 ml ang iyang capacity for this. So that they can be in a glass form, the quartz and the glass, and also there are already plastics disposable now that are available that you can really just throw it and dispose it if the the transparent sides are really already damaged and you can really it is not reusable now. So and then there are also model class of a spectrophotometer that have multiple sample chambers in which dili na ka mag you will not having a hard time that you will you place your sample, you place your blank, no, your blank. There's a sample, there's a model class, a spectrophotometer na say blank chamber, na po siya sample chamber in which dili ani mo siya kuhaon imo blank chamber. Yeah, you will not really get your blank chamber because you just have to place it into the blank chamber. And then, ang sample chamber na imong puli-pulihan o kanang butang. There should be one sample at a time. But there are already advanced spectrophotometer classes has many sample chambers in which ang machine or ang equipment na lang ang mo tuyok-tuyok that tuyok asa ang mamurid na siya ang absorbance about 10 cuvettes. Yeah, 10 cuvettes. You have this 10 cuvettes already. So you don't have a hard time just placing and getting your cuvettes one at a time. So you just have to load it ang napulo ka buok and then you just have to wait for how many for a few minutes and then you can now get the result. It will then just be reflected sa computer. So that's how these spectrophotometer works. Okay, so based on my experiences class is that I use this not just in the ethanol determination but also in the concentration of your in the bioprocess, diba, sa microorganisms using optical density. So, 300 to 60 nanometers na siya ang kanang wavelength sa yeast, sa bacteria that you have to identify its concentration if it's already viable for the growth of your microorganisms. Ganun. So, the concentration of your cells. And it is used for the optical density determination, the microbial growth curve. Yeah, you can really use the optical density in order to plot your microbial growth curve. Pangalawa, you can also use your this spectrophotometer for the free radical scavenging activity of these plants, of these plants, those bacteria that has free radical scavenging activity. So there are chemical reactions also, chemicals that are involved in order to achieve a chemical reaction. So I have also experienced using this spectrophotometer in the lab. The vitamin C concentration, you can also use that one for the different compounds, and also in my undergraduate thesis, the lovastatin compound, I use it as a determination for the concentration of the certain compound. So there are many uses plus of spectrophotometer. So it's very basic that we have to familiarize it and we have to know its principle. Okay, so I will now go back to the Okay, so the absorbance of this ethanol is 578 nanometers in the UV visible spectrophotometer. So the standard curve class, muna siya class ang imong results sa absorbance sa imong 2%, 4%, 6%, 8%, 20, 10%, and 12% that you have to plot it against your absorbance. The concentration, the x-axis is the concentration and the y-axis is the absorbance, in which you have with this standard curve, in which these are your basis for extrapolation or interpolation of the concentration of your unknown sample. So it should be that the linear regression of this standard curve is 0.97 to 0.99, the r squared, the linear regression or the R squared, it should be linear, okay? So that's the standard of determining the concentration of the unknown sample. Since we didn't conduct this face-to-face, so exciting sana ito na i-conduct class, but then due to limitation of our movement. So in your lab reports class, you just have to discuss all the principles of the spectrophotometer. Okay, so please take note of the content of the discussion. So the definition of spectrophotometer, and then also in the discussion you have to explain the uses of this activated charcoal, the potassium dichromate, and then also the why is it should be diluted, class, ganon, and in preparing your what are the best practices in preparation of your standard curve. Why is it necessary that you have to put your standard curve in determining a known concentration of sample? Why is it that there is a water bath at 62 degrees Celsius for 20 minutes? Ganon. So why it should be cool? Ganon. So that's the flow of your discussion classes. We have no results to be discussed, so the principle na lang of this spectrophotometer. So I guess class, that's it. Okay. So do you have any questions?
None so far, ma'am.
Is it clear sa inyo ha? This will be the last exercise.
Yes, ma'am.
Okay. Good. So we still have time. So I will now proceed class. Okay. Can we have a break for 5 minutes muna because I will proceed to the lecture. I will give you a lecture class para makaapas ka sa lessons. Sa doon na po ang final. So, kabalo na mo ano sa inyo final exam?
Wala, ma'am.
December 13 to 17. 13 to 17 na inyong final examination. So, ano siya ang inyong last exercise. So, I will prepare that next week we'll now have your final exam. Or do you want sa 13 to 17 na lang?
Okay ra, ma'am.
Sa next week or 13 to 17?
13 to 17.
Okay, sige. Okay, we'll rest muna for 5 minutes, then we will proceed now to the lecture. It's the lesson 1. Then the rest of the lessons I will now be recording. Okay, class. So I'll now proceed to the lecture part. Class, can you see the full screen?
Yes, ma'am.
Okay. Okay, so this is now the module 4 about enzymes. Okay, so I guess in INBT detect 111, you have your proteins and nucleic acids, right? So it's already tackled. Proteins are enzymes. So also you have encountered this in detect 22. So it's just a um, refreshment of the lesson from the previous topic. So this is the pre-test class in which you have to answer it in the VSUEE. It is not graded, but you just have to participate in your answering your pre-test identification. So, okay, so I'll just read through this um, questions or statements. You have to input your answer in the VSUEE so that can really we I can really saw your answers and also evaluate afterwards if you have really um, understood the topic. Okay, so the first statement is these are protein compounds that catalyze all the chemical reactions of living things, the plants, animals, and microorganisms. So the second part is that this technique is achieved between a functional group on the enzyme that's essential for catalytic activity and a reactive group on the solid water insoluble support. Now, in this type of mode of operation, the enzymes are poorly stable and hard to recover in such systems, and it leads to low productivity. Okay, so actually, this is the whole part of the module. So it is a four statement. It is first downstream operation that is to be done after fermentation, and it can be done conventionally by centrifugation or dead end filtration. With the last statement, you have to identify this method. What is this method that has been traditionally used as initial purification steps because they are simple and readily scalable. Okay, so you have to answer it in the VSUEE class. So for the lesson 4.1, the definition and uses of enzymes. So at the end of this, you will have to define what are enzymes, to understand the properties of enzymes, and the last one is to determine the uses of enzymes in the industry. So as a trivia class, this is Anselm Payen. He was the one to first to discover the enzyme in which this is the diastase. The diastase was the first enzyme discovered, and it was extracted from malt solution on 1833 by Anselm Payen, and also he has a colleague, Jean-Francois Perzo, the chemist at a French sugar factory. It is used to convert these malt solutions into simple compounds. Okay, so this is just a trivia. So by definition class, what are enzymes? So protein compounds these are protein compounds that catalyze all the chemical reactions of living things. It includes plants, animals, and microorganisms. We are surrounded by many enzymes, and they contain mainly amino acids that make up a protein, while some have non-protein components or prosthetic goods. Okay, so all enzymes are proteins, but not all proteins are enzymes. There are many kinds of proteins. You have chaperone proteins, you have structural proteins, so enzymes are just one of that aspect of proteins that is a lot of it to, to be investigated. So these are really complicated and complex uh, structures and also has a sophisticated um, uses that we have to discover, and there are many enzymes that are yet to be discovered. So they can act in an independent manner when extracted from the producing organism of their source. So the bacteria produces enzymes, and also plants have enzymes also. So we have in our body, eukaryotic cells have enzymes also. Prokaryotic cells have enzymes. The protein molecules, these are protein molecules uh, produced by living cells and act as biological catalysts for most biological reactions. So these are catalysts. It's an aid for for reactions, for biological reactions. So its catalytic activity and specificity can be attributed to the tertiary structure of protein. So I have already discussed with you the different structures of protein, the different uh, the primary structure, the secondary structure, the tertiary structure, and the quaternary structure. So the catalytic activity and specificity of the enzyme is connected or linked to its tertiary structure of the protein. Now, the reactant upon which enzyme acts on is referred to as the substrate. So when we say that an enzyme can degrade for example, the invertase. Okay, invertase can really degrade sucrose. So the sucrose will is the substrate. Diba? Ang sucrose is a combination of fructose and glucose, right? So with this invertase, so the enzyme invertase will really degrade or convert this disaccharide into monosaccharides, fructose and um, glucose or galactose. So, yun, they have also been a major component in many ancient human activities, especially food processing, well before their nature or function was known. So, they didn't even know that the traditional people that they can really the enzymes are present in their food and that aid for that are produced by the microorganisms that aid for the food spoilage and also the conversion of this rice into liquid form. That there was a wine fermentation that happened. So they didn't know much about enzymes because, of course, they are traditional people, and I guess they have lack of knowledge and also curiosity how that works. No, so that's it for the enzymes. So I have already discussed with you also, and also you have learned about the levels of protein structure. So the tertiary structure is the linked, is correlated with the specificity and also the catalytic activity of the enzymes. The folding and the coiling due to the interactions among the R groups and between R groups and surrounding water. So these are really that makes it unique to its use and also the catalytic activity. Now, we all know that proteins are the major backbones of all enzymes, and all enzymes are proteins in general. So, but Proteins are not just attachments, but some enzymes have a covalent attachment with carbohydrate groups. Okay, these carbohydrate groups may play no part in the enzyme's catalytic activity, but it will contribute to its stability and solubility. So that makes the enzyme more stable and soluble. So we have also cofactors and coenzymes. No? So moreover, metal ions known as cofactors and low molecular weight organic compounds known as coenzymes is also existing. No? So sa PCR class, isa ganito ang kanang enzyme that is responsible for the synthesis of a new strand of DNA in the PCR technique. Ang sa ganito siya,
DNA polymerase, ma'am.
DNA polymerase. So, unsa nga specific polymerase yung ginagamit sa PCR. Taq polymerase. Yes, diba? Taq polymerase. As you can see, class, as you have, sa components sa PCR class, naagid na diha ang usaman diha ang cofactor, ang magnesium chloride. So you have your template, magnesium chloride, the buffer, and also the DNTPs, which are your nucleotides, ganon, and also your the NTPs and also your primers. But what I'm trying to say that the magnesium chloride, which is the Mg2+, this is your cofactor that initiates or that helps your Taq polymerase to function for its synthesis of the new strand of your DNA, for the complementary strand of the single-stranded DNA. So this is one of the example, which is a metal ion, so the Mg2+. This is a cofactor. Okay, so that's the example of these cofactors. Okay, and also cofactors and coenzymes are essential for the stability activity of the enzyme. Okay, they are susceptible to detachment, but conditions for retention should be critically observed to prevent loss of activity and denaturation. Always remember, class, that enzymes are really sensitive, temperature sensitive, others are light sensitive, and also pH sensitive. It should be maintained at a particular pH. So prone to denaturation ang ubang protein. Okay, just like with your Taq polymerase, and also if you are using this endonuclease for your molecular experiments. Okay, so you have to observe proper usage of these enzymes, because when we are talking about molecular experiments, we should use these enzymes so that this will be for the degradation of a certain strands of a single DNA, single-stranded DNA, ganon. So if you use cloning, if you want to restriction digestion, so you have to use enzymes, and enzymes are very, very pricey. Okay. So what are the properties of enzymes? So number one, they are really specific. Okay. So in a particular compound or substrate, they will just only react with a one given type of substrate. Okay. So when we say amylase, okay, amylase is an enzyme that converts the amylose into different disaccharides. Okay. The enzyme that breaks proteins will not break down fats. And for example, the lactase enzymes only break down lactose. And amylases only break down starch into disaccharides and trisaccharides. Now, the alpha amylases are present in our saliva, secreted by the salivary gland. Okay, so that's how specific the enzymes are. Enzymes are also affected by extreme temperature and pH. And each enzyme has an optimum pH and temperature at which it works best. Okay, because of the component structure, class, diba? We have an NH plus, and also you have COOH, negative COOH part of your amino acid structure. So it is very susceptible to change of pH and temperature, and especially also that the R groups have many different stability. They have different R groups depending on neutral. They are also negatively charged. They are positively charged, and so polar, non-polar. So ganon. So enzymes are not affected by the chemical reaction that takes place, and enzymes are affected by poisons such as toxins and heavy metals by a high concentration of salts. No, so it is not affected by the other chemical reaction that takes place, but it is just affected by poisons such as toxins and other proteins or enzymes and heavy metals by a high concentration of salt. So it is prone to denaturation. So that's it for the depending sa enzymes, if they are really tolerable at a certain concentration of salts, no, there are microorganisms. These are halophiles that can grow or can thrive in very high concentration of salts. No, so it's just it depends upon the niche or the environment that the microorganisms, talk about microorganisms, and you have that microorganisms contains many produces enzymes for their metabolism. So we have also key roles of enzymes. They have vital roles in numerous biotechnology products and processes that are commonly encountered in food and beverages industry, in the cleaning supplies, your laundry soaps, your detergent powder. It has enzymes. You know, these are proteases, amylases that really can remove dirt and stains. Okay, clothing, paper products, transportation fuels, pharmaceuticals, and monitoring devices called biosensors. So the most frequently used enzymes in biotechnology are hydrolases, which catalyze the breakdown of molecules. So we have also another role of enzyme that it can display regio and stereospecificity properties that has been exploited for symmetric synthesis of racemic resolution. Okay, so this is the stereospecificity of enzymes, that it is alpha amylase, no, the alpha 1,4 link to local starch, so it is not, it will not convert or it will not catalyze for the beta 1,4 link, no, for the cellulose. It is just only for the alpha 1,4 linked glucose, which is in starch. Okay, so chiral selectivity is employed to prepare enantiomerically pure pharmaceuticals, agrochemicals, chemical feedstocks, and food additives. So because other chemicals are levorotatory and dextrorotatory, enzymes can catalyze reactions specific to the rotation of the desired product. So that's how the enzymes are really amazing, that it's really highly specific. Okay. Another role is that it has expanded at a rate of about 10% annually. Microbial enzymes have primarily substituted the traditional plant and animal enzymes, and most of them are produced by RDE technology. So in the juice industry class, no? So the enzymes that are commonly used in order to have a to have a clear, so that your juice that are already found in the market has a clear solution in which clear na siya, not really pulpy na, but there are also juices that has all, like Minute Maid that are pulpy also, but we want to have a clear solution, not viscous ba. Ganun. So they use pectinates in order for the pectins will be dissolved and will be degraded. So ganun ang pectinates ang ilang uh, gina-add for them in order that the pectins will really be converted and will really be dissolved. So that can really have a kahit mapakla ramang pud kaayo class kay kung sa imong juice na kanang mga natural juices gid pakla ra pud kaayo nga ano bang ang imong mga mga pectinates ang mga pectins kay dili siya ma-dissolve. So mga siya pectinates ang gamit ana class. So next, for the DNA technology, it has been employed to modify substrate specificity and improve enzyme stability properties for increasing yields of enzyme catalyzed reactions. So one of the example of this is the PCR and Taq polymerase. It is very specific to the synthesis of the complementary strands, and also there are endonucleases, restriction endonucleases, which can cut a specific base pair of your, for example, plasmids. If you want to do cloning, diba? You have also the DNA ligase that are already can be purchased from the different molecular companies. So there are a lot of molecular enzymes that are really useful in the recombinant DNA technology, diba? So ang restriction endonuclease will cut the specific portion, the specific base pair, no? So while the DNA ligase will really seal the the gene of interest in your plasmid and also the gene of interest, okay? So that's how, and there are also many enzymes that are really useful for the synthesis of the DNA templates, no? So there are a lot. So you will encounter it sa BTEC 134 next sem, no? So another is that enzymes are also used in metabolic engineering of cellular metabolism to increase the yield of fermentation products. These are environmentally friendly. They work under moderated conditions of temperature, pH, and pressure. Their catalyzed reactions rarely form wasteful side products, and the proteins themselves are biodegradable and generally pose no threat to the environment. So I have also read sa mga articles that during the oil spill, so they use microorganisms that can produce enzymes that can convert these oils into that can degrade the oil, no? So decrease the concentration of oil in the seawater. So mana siya class. These are environmentally friendly. So okay, so there's another one that how about when we compare the comparison and enzyme catalyst? Okay, comparison of enzyme catalyst. Now, an enzyme catalyst is highly specific and catalyzes only one or a small number of chemical reactions. The rate of an enzyme catalyzed reaction is usually much faster than that of the same reaction when directed by non-biological catalysts. Synthetic, purely chemical reaction. Now, only a small amount of enzyme is required to produce a desired effect. The reaction conditions should be observed, in which it has temperature, pressure, pH, and so on, in which for the enzyme reactions are very mild. So the enzymes are comparatively sensitive and stable molecules, and these are the drawbacks when we really when we use enzymes. No, it will require care in their use. So I guess I will end this class. So next recording or the video recording, I will continue with the classification of enzymes. Okay. Do you have any questions?
None so far, ma'am.
How about the others? Is it clear?
Yes, ma'am.
Okay, so please, I will just post an update in our group chat and also the VSU EE for further announcements of your projects and also your examination. So your classmate said that it's okay with December 13 to 17 that you will take your final exam for the lab, and it's okay for me. Okay, so if there is no more questions, I guess I will now dismiss this class. But wait, first I will. Can you open your camera, class? It's okay if it's okay with you.
Yes, ma'am.
Are you in a good lighting and also dressed properly? Ganon. So that you can really take a screenshot. Okay. How about the others? Maka-open mo sa inyong camera. Thank you, Jasmine. How about Mizuki, Ricky, and Liza? Okay.
Ni ako, ni.
Sige. Okay, I will now screenshot. Okay, 1, 2, 3, smile. Ay, wait, ako muna. I-off akong camera. Okay, 1, 2, 3, smile. Okay, thank you so much, class, for joining. God bless. Bye-bye.
Okay, ma'am.
Good evening, ma'am.